not anything they can treat, but the doctor was like, oh, this was like 50, and so he goes, you know.
I don't know if there's one. Is there a little? There's more. There's like one here. Is that? Yeah.
It's like me, I run a lot, and if people talk about knee pain, I'm like,
That's cool. I'm glad you.
Yeah. So he's like, wow.
Thanks. That works well.
But
It's
you
like a
really
freaking
talk
job
about
talk,
knee
man.
pain.
It can be like me ashamed to work on my shit. We talk to us compared to that.
That's what I'm glad we don't.
No, but it really, like, everything is like, should I go back? Should I wait another week? And it's like,
When you think of
Because,
portfolio, what are the axes there? I think I was like lost specific reading.
yeah, I don't know.
Honestly, the portfolio effect confuses me so much. I get so.
Well, I think.
I don't understand the difference between a portfolio
I'd have
effect
to
and
go
insurance
down for
effect.
it.
I need to read
I
that
don't,
one
I don't
over
read
again.
that, yeah.
I need to read that.
I actually didn't realize
My parents were.
Apparently there is a subtle difference. There's a really useful table in that page, that the row
yeah.
paper
Like, no,
thing
you
you was
can't-
like 2020.
It's like what I do.
The insurance effect is more about the effect of diversity.
Right.
But I'm like,
Overall
I'm like,
biomass.
Yes.
I don't go running.
In my mind,
Wow.
portfolio effect
Yeah.
is the relationship between average population.
If I don't have enough time,
Yes.
because I don't.
Like basically, like his thing is
Yeah,
exercise until
maybe, yeah.
failure,
That
so
sounds-
like that.
People. Yeah, I always thought that, but it was like, it was definitely an averaging, right? It's like you've got these individual things, and it's like, what does the overall portfolio do?
But so.
Yeah. Oh, I don't know. Oh, he keeps telling me he's like.
Yeah, I must have read that paper like 3
"Oh, I
times.
need to go back. Like,
Sounds like
the
it.
answer's fail. And then he's like, my arm.
Maybe you should join and then you get motivated.
Yeah.
Nice talk, Sam. One of the best I've been.
You're biased.
I'm not. It's not only about the data, it's also about how people present it.
Yeah.
Yeah.
Oh yeah, hopefully it will be a job talk at some point. Yeah.
It wasn't already.
I
Description of it.
haven't actually presented this yet. I mean, I've presented it at a bunch of Japanese universities, but not officially as part of this. Yeah.
I had like 2 slides at the end about like future research.
Yeah. Yeah. To be fair, a lot of them are my friends that are not.
They're not coming back.
ecologists. I suspect a lot of them might not be coming back. Dave's lab should come back.
There were a few sitting chatting outside, so they haven't just totally abandoned it. It's Friday.
You're right, yeah, it is Friday, yeah, that's true.
Try and be energetic, enthusiastic, and quick.
Those are all good properties.
Can you just throw out shots of espresso?
There's some interactive element, throwing things that just throw something at Mike when he falls asleep.
Yeah. Oh, really, I'm proud of myself making it this far.
Yeah.
Must have been the quality of the talk.
Good grade for the talk.
Thank you.
Good grading for the talk.
I was going to introduce, it sounds a bit dramatic using all of these different things. I was going to say, say you're director of, is this true? What is this? The research priority program. I was going to ask you about that. It's like a cluster.
It's a group of professors or research groups in the university that come together to work on research. That's funding from.
Yeah.
Come on, people, we don't have all the.
Yeah, however you.
It's good proactive. Like integrated.
Yeah. It's not just even.
Yeah, it's obviously.
I hate it when people don't respect each other's thoughts.
Come back in here.
In fact, yes.
Then I noticed that who was the card reader.
Well, we do have to tell them that next one. Yeah.
No, I was up for some stuff.
Maybe just look forward. Yeah. I have never had one of those before.
Is this working? Is it? I can't hear myself.
Yeah, I was in it.
Good.
It's not a very big room.
I like to have a bit of amplification because sometimes I get a bit quiet. So.
Okay, I guess I can share.
Yeah, I certainly am not.
I just you want to get to.
Hello. Hi, everyone. Thanks for sticking around for the next talk in our series. I'm going to introduce Professor Owen Petchey. We had this conversation the other day. We can't exactly remember exactly when we first started talking to each other, but we certainly got in touch a few years ago to start thinking about response diversity together. And since then, yeah, I mean, we've done a couple of bits and have organized this thematic sessions, this thematic, it's called program. So yeah, I'm delighted to introduce you to Professor Owen Petchey, who's from the University of Zurich, the Department of, I always get this right, Evolutionary Biology and Environmental Studies.
Very good.
Thank you. And he's also the director of the Global Change in Biodiversity University Research Priority Program, which is a bunch of interdisciplinary people who do work on biodiversity and global change. And I'm going to now pass over to Owen to talk about understanding and predicting the stability of ecological systems. Woo!
Thanks very much, Sam. Thank you very much for coming along to listen. Thank you very much to, well, Jonas isn't here now, but organisers of the thematic programme and the theoretical sciences visitors programme. I've been here since Monday, and I'm just about over the jet lag, I think, but I can definitely say this is the first time I've ever given a talk at 7 a.m. Which is what time it kind of is for me. It's also 3 p.m. on a Friday, so I need to keep this a bit lively and engaging. So let's get going. It's great that Sam spoke first because I don't have to talk about a lot of things about stability, but so I can whiz through the first few slides and then get on to talking about more ecology in buckets. Small buckets. I'm going to talk about 2 studies that we've done in the research group over the last 5 or so years. Just focus on 2 really, and go into a little bit of depth about those. One is in buckets and one's in computers. You get ecology in buckets and ecology in computers, but just little buckets. So what is stability? Why are we concerned with stability? The introduction, and then these 2 studies. How does biodiversity affect overall ecosystem stability? And what is that overall ecosystem stability, of course? I'm going to kind of describe that. And then how does biodiversity affect when a catastrophic change occurs? And this is related to one of the slides that Sam talked about, which is people are talking about how he's talked about how response diversity relates to this temporal variability, that kind of stability. This is concerning a bit about how response diversity can affect the movement of this ball in the landscape from one basin to another basin. So we'll get onto that kind of stability. Stop me at any point if something's not clear or you just want to ask a question. There's few of us here that we can do that quite safely, I think. So what is stability? We've already had a good introduction to this, but I'm going to change it a little bit. I haven't got Stretch Armstrong with me. I've not been able to find one, but I do have something that's a little bit like that. It's a rubber band. And I'll try and demonstrate these 4 components of stability with this. One of them is resistance. And so what I'm going to do is apply an external force to this, a bit like one could think of a temperature change affecting a community. I'm going to stretch it outwards. And the amount of force I put on then causes it to move a certain amount. And if this one's quite resistant, actually, I've got some others that are thinner rubber, and they move further for the same amount of force on them, and that's resistance, how far it moves when I apply force. And then resilience is how quickly it goes back to where it was before, right? This is rubber, so it goes back very quickly. Variability. So if the environmental forcing is kind of continuously varying, then the length of this changes continuously as well. And that's due to both its resilience and its resistance, how fast it's returning, and also how fast it deviates. And then there's persistence as well. This is persisting in its configuration. It is still a circle, whatever I do to it. In contrast to, well, this, and then you might think, well, what kind of stability properties does this have? It's got a tissue from my office. It's actually quite resistant, right? Because I'm stretching it and it's not, actually, there you go. I was stretching it up to a point and it wasn't changing its configuration. So it has a little bit of resistance, but then actually you can see it's not persisted. So very different stability properties, low resistance, virtually, I mean, we can't easily measure the resilience of this because it just broke very quickly. Now, resilience also has another meaning, which is how much one can push or force the system without it changing to another configuration. We didn't experience the limits of this moving to a different configuration, but we did with this. So this is now in an alternate state. Not necessarily, well, actually, yeah, it's in an alternate state. So that's how I think about stability a little bit. Analogies are useful up to a point and then they break down, and then thinking about how they break down can be useful as well. Be happy to talk more about those kind of analogies. And in general in the group, we're interested in kind of what kind of substances with what kind of properties are ecological communities like? Are they stretchy and compliant, or are they stiff and very resistant? And what are the features of the ecological communities that make them either rather resilient and stiff, or very or very easily stretched and snapped, if you like? So is that species richness? Is that something that affects the stability of the community? I'm going to talk about that. And also response diversity in the last part of the talk. This is not new at all, as you know. This is a paper that Stuart Pimm wrote back in 1984. So stability goes way back, and he talks about those 4 components of stability. And also that when we're measuring stability, not only do we have to choose which of these we're looking at, or which combinations of these components of stability that we're looking at, we also have to think about the stability of what? Will it be of individual species abundances, the species composition, of the total abundance of a trophic level? And so this already makes quite a lot of combinations. If we were 12 combinations of things here, and then we can also think about stability in different types of changing environments, a press perturbation or a pulse perturbation or continuous variation. So we very quickly get to a situation where talking about complexity and stability, or talking about just stability, is really complicated and difficult. We need to specify a lot of kind of context in order to be able to talk about it in a sensible way. So it's multifaceted, but actually simple messages often have greater influence. So we want to be able to communicate this simply, our message simply. One of the questions then arises is, can we do that without losing too many details for it to be useful? I'm going to talk about that in the first empirical example I go through. So stability is complex and multifaceted with many measurable components. That's not easy to communicate, but we're going to give it a go with this overall ecosystem stability concept. Why are we concerned with this? Well, Sam's already talked about that. Maybe it's really obvious to you. Let's just reinforce that a little bit with a study that I and some other people here are involved in, led by Donohue. And what we did there is look at various kind of white papers, reports, and documents by organizations, and these documents such as the Aichi biodiversity targets, documents of the Food and Agriculture Organization of the United Nations, the Organization for Economic Cooperation and Development, and IPBES, And we went through those and looked for words that we thought were associated with stability, already directly related to stability. So in the middle here are the words we found. And we can see resilience several times. We see safe ecological limits. And then we see words that we don't actually see over here, which is the ecological, the ones that are common in ecology. We see integrity, conserve, sustain, and sustainability. But I think that we can in some way, and we argued that we can in some way relate these terms, like variability, for example, to safe ecological limits. And so there is actually a mapping, although it's rather loose in some cases, from the work that we do in ecology, these foundations, the work that we do in ecology through to these documents and kind of sentiments to affect policy. That's what a lot of these are about. Let's try and affect policies in a way that are going to lead towards a more sustainable and a more stable world. And so we were arguing, and many have, that the work that we do here can have direct influences on these documents, and thereby we can try and affect policy towards being more sustainable and more stable. So I think it's really important that we do that work in ecology, but also that we work on trying to translate that as well. Another reason why stability is important, or I think or why it's research maybe, is because it's interesting. It's not simple. And sometimes we find opposite patterns. So going back to the work that Charles Elton did, he suggested that more diverse communities are more stable, or actually less diverse communities are less stable. They're more likely to be invaded by pests. MacArthur suggested the same thing, that more species, that's number of species, leads to greater stability. And his argument was because in a more diverse community, there can be more routes for energy to flow, more different pathways for energy to flow. In a way that creates insurance, because if any of those single pathways are lost, then many different pathways still remain. And of course, Bob May came along and said, no, that's all wrong. But this was a different kind of stability. This was whether a system returns to an equilibrium when it's disturbed by a very small amount. And so in that situation, more species and more interactions caused lower stability. And that's what's shown here, except in this work by Stefano Allesina and Tang, it's actually the type of interaction is very important. So communities that are in this region here below this kind of line are stable, and the ones above are unstable. And you can see the predator-prey communities have a much greater region of stability than those with mutualistic interactions or competition, or just random interactions. So it's really important to know which kind of interaction there is. And this body of research is huge now in ecology. So it's really interesting, difficult and challenging field of ecology to get involved in, which I think it makes it really, really quite a nice one to study. So not as only is it important, but it's interesting. All right, let's get on to the first study, the one with the very small buckets. How does biodiversity affect overall ecosystem stability? That's what this one concerns. And this is a project that we did back in, I guess, 2016. I think we actually did it. And then the first publication came out in 2018. And it was by a team that was composed of my group and also the group of Florian Altomat, who is just there. That's Florian. And it was led by Frank Pennekamp. So he was the lead of this, and he's the lead author of the publication. It was a lot of people because it was a lot of work. The question we were asking is, how does stability components vary along a diversity gradient? And how does overall ecosystem stability vary along that diversity gradient? To answer that question, we did an experiment. Those are the buckets. these are jars of a nutrient media with bacteria that are fed upon by some combination of these 6 species of ciliate. And we assembled communities with either 1, 2, 3, 4, 5, or 6 species. So each jar has a separate community in with different levels of species richness, and there were different compositions within each species richness level also. And then each of those jars also experienced either 15 degrees C, 17, 19, 21, 23, or 25. So it's constant temperature, different temperatures for some of the jars. So it was a two-way factorial experiment with species richness and temperature. With the compositions and with the replication, it was 690 of these communities, sampled 19 times over 40 days, resulting in a hell of a lot of samples that had to be processed. It was the biggest experiment that we've ever done, and that's why we called it the BEEP, the big ecological experiment about prediction it was at the time. So to get the number of individuals in each of the species at each time point, we took some liquid out, put it on a slide, and then put it under the microscope and videoed it. And then we used some software that we'd written, go to the next slide, to separate the particles that are moving around. And that happens to be the protists that are moving around from the background, and then identify them using some a classifier, kind of machine learning classifier that we'd already trained on the monocultures. So here you can see a community with just 2 species, the red ones and the purple ones. And so what we ended up with after the sampling was a lot of videos, and then we spent actually quite a lot of time processing those videos. The results I'm going to show you are only use the number of individuals, but the nice thing about this computer vision system that we designed is that it also gives size of the individuals, various other traits, swimming speed and such like. So we can do analyses with that if we want to. Say something else about that. Oh yes, all of the code is open as well. So it's written in R, also in ImageJ. It's available, and we're quite happy to help you use it if you'd like to try and do that for some of your videos. The main thing is that one needs a video with very good contrast between background and foreground. All right, so from that we got many time series of population dynamics. From those, we computed 2 measures of stability. This one, the temporal stability of total biomass fluctuations. That's the mean of the total biomass through time. And that's the standard deviation of the fluctuations of total biomass through time for a jar. So each of these data points is 1 jar, and how much total biomass has fluctuated through time. The top is fluctuated not much, and the bottom is fluctuated a lot. So it's flipped in a way. So it's high stability, low stability. And this is what we expected to see, positive relationship here. So increased species richness increases stability. Yeah. That's what we expected to see because of the insurance hypothesis. And this is another measure of community stability, it's resistance. You see that's the change in density, total density of the community, and it should say also that per degree C. Okay? So it's the sensitivity of total biomass to temperature. How much does the total biomass of the community change with 1 degree C change in temperature? You can see actually the resistance goes down when species richness goes up. Those more diverse communities, their biomass, the total biomass of those more diverse communities is more sensitive, it changes more. We did some analyses and found that the first result there about temporal stability is due to complementarity, so there's higher total biomass in the more diverse communities, and some statistical averaging. It's just having more randomly fluctuating time series leads to more constancy and not increased asynchrony. And on the right-hand side, we think that's due to lack of response diversity. All of these species responded negatively to increasing temperature. We didn't have crossing performance curves in this case. So we have these opposite effects of diversity on stability. And then, so that's a bit frustrating, right? Because the answer then is, well, it depends. And I really don't like it when we keep saying in ecology, well, it depends. It depends. So let's try and come to an answer that is not that. The punchline is, I'm sorry, but we can't. So one way we can try and do this is to come up with a measure of overall ecosystem stability. Overall ecosystem stability. How can we do that? Well, we could just simply add the two measures together. We're going to have to standardize them first, though, because they have different units. And if we standardize two linear relationships like that and then add them together, what do we get? We get a flat line. Right? They cancel each other out. It can't be anything else. And so I thought, well, that's not particularly interesting. I went to some of the research about ecosystem multifunctionality to see how they created measures of ecosystem multifunctionality from individual measures of ecosystem function. And something that they do is they do another step. So they do the standardization, which I think you have to do because they're in different units, but then they also do a non-linear transformation to go from the standardized stability component value up to the converted. I'm not sure there's a really very good reason for that, but that is something that they have done. So we thought, well, we'll give that a go as well and see what happens. I'm going to switch now. So here, this is what you just saw on the presentation. It's the standardized stability component, like resistance, converted into the converted version. And what we have, there's many parameters for this curve, but one of the key ones is where this threshold is. Is it here, or do we move it towards this end, or do we move it towards that end in the conversion? And one of the things, and what this is the sum of the two stability components after they've been standardized and converted. What this is on this axis is each of the data points with that conversion done, each of the communities and the line fit through that. And this is just the mean for all of the communities at a particular richness level. Okay, no relationship here. This is when the threshold is right in the middle of the standardized values. But if we change this threshold, we say, well, actually, we're going to make it so we need higher stability, higher standardized stability to actually get, lower, let's say lower standardized stability to get a high converted. You're going to change the threshold to be at a lower value. And we get this hump-shaped relationship between overall ecosystem stability and richness. But if we do it the other way, go to this end, we get a U-shaped relationship. Now, this tab here shows why it happens. And I'm not going to go through that right now because I think you would all fall asleep. And probably I would as well. It's quite difficult to explain. The thing that's really obvious is that this choice here is completely changing the diversity stability relationship. And actually this is where it's still not satisfactory because the relationship depends on this choice. So it still depends. We haven't gotten anywhere really by adding these things together. You know, maybe somewhere we can get some information that would say, actually, we should put the threshold here. So we could ask some experts perhaps, and they would say, we can explain this whole thing to them. And they'd say, we'll put the threshold there for this reason. And then we would have a more kind of objective or reasoned stability diversity relationship. But that is, let's see if I can get back to the presentation. Yeah, that's where we got to with that research. And maybe you remember the title of the paper, biodiversity can increase or decrease ecosystem stability. And it's increasing or decreasing because that depends on the component that one chooses, temporal variability or resistance, but it's also increasing and decreasing because sometimes you can get hump-shaped relationships and sometimes you can get U-shaped relationships. So that is kind of somewhat satisfactory, but also there's still a lot to be done there. And I think the focus actually, if someone wants to make or use a measure of overall ecosystem stability or multifunctionality, we've got to really focus on those conversion functions, how we choose where the threshold is, how we weight different stability components differently. And that's something that has not been talked about a lot. It's very similar actually to the work that I've done on functional diversity, measuring functional diversity, where one takes many traits and then puts them together into a measure of functional diversity. Many people for a long time talked about, well, should we use this measure of functional diversity or this one, and didn't so much talk about trait weighting and trait conversion, trait transformation, whereas actually that's the most important part of it, I think. All right. Any questions on that? I'm going to move on to something quite different. I don't know how I'm going to do this. On a Friday afternoon at 3:30 or 7:30, this could get a little bit heavy. So let's see how it goes. I'll try and keep it a bit lighter because there's lots and lots of equations that I'm not going to. They're all on one slide though, and I'm not going to explain them. So you can relax. All right. This is work that was led by Romana Limburger, a postdoctoral researcher in my group. And it's another example of us doing projects in the group with many people in the group working together on those projects. It's a way that I really like us to work together. And this is the kind of context for that. We have a environmental change, like temperature, that's happening gradually through time. Then we also have an ecosystem state, and this is the external condition on the x-axis. It's not time here. We see that here the ecosystem state changes gradually as the external condition changes. In this situation, there's a threshold. So once the external condition, like temperature, gets to this point, then there's a quick change. And then it smooths out again. And then there's this situation where there's a discontinuity. We go along here in a supposedly healthy state. We get to this point, a quite high temperature, and it falls off this cliff down to the degraded state. And then if it moves back, it doesn't instantly go up to here, but rather it goes along in this degraded state before it flips back to the healthy one. That's a system with bistability. So in that region between there and there along the x-axis, the external condition, the system can be either healthy or degraded depending on history, where it's come from. And Sonia Kefi and others made this very nice paper, and the figure, which I took from there. And the main mechanism for causing these discontinuities is a strong positive feedback loop, in the system. I'll explain that a little bit more when I come to the specific system in this study. And then Vasily Dakos and others came along and said, you know, the models, most of the work on these systems with biostability has involved very simple ecosystem models with, for example, one functional group or two functional groups and no evolution and no diversity within those functional groups. So every organism within the functional group is exactly the same. And he came along and said, well, what happens if there was evolution? If the organisms in a functional group could evolve? And they didn't come up with the answer, but they come up with the two possibilities. The red line is with evolution, and the black is without. And if the environment changes along here, environment stress E increases, without evolution, it would shift at this point, whereas with evolution, it could shift at this point. And so evolution, having evolution, would cause an increase in resilience. If we were measuring resilience as the amount of environmental change before the system flips, an increase of resilience of this amount. Or this could happen. It could cause an earlier shift, and then there would be a decrease in resilience. They didn't come up with the answer of what should happen. Oh, sorry, what would happen in any specific case. They just were saying what could happen. And so I thought, well, a few things happened. And then I thought, I'll give it a go, see if I can do this for a specific system. And then I thought, well, I'm not very good at modeling evolution. I've never done it. So I'll do something that's a bit easier and that I have done before. And that's modeling trait diversity. And so that's what we had a go at. And because of those things that happened before, I got interested in this paper before that and did a reproduction of this paper. So I'd already been playing with this, the model in this paper, which is about microbial community dynamics. So I already had a reproduction. I thought, oh, well, that's a good starting point then, because I won't have a lot of work to do. Well, that was wrong, because there's always a lot of work to do, even when you have a good starting point. But let me explain this system. It's got basically two states it can occupy. An oxic state, so high oxygen levels, and that's when it's dominated by cyanobacteria. They produce oxygen, and it's got an anoxic state, and there it would be dominated by the sulfur metabolizing bacteria, which produce reduced sulfur, hydrogen sulfide, and that is toxic to cyanobacteria. Okay, so the red is inhibition, red line is inhibition. Whereas cyanobacteria photosynthesize and produce oxygen, and that inhibits, it's toxic for the sulfur bacteria. So although this looks really complicated with lots of different parts, you can just basically see this loop. Look at that loop. And it's the loop with mutual inhibition. Cyanobacteria inhibit sulfur bacteria inhibit the cyanobacteria. And that results in this system being able to display alternate stable states. This is just a simulation. Actually, I think these are the results from their publication, Bush et al.'s publication. And they specify particular parameters and particular environmental conditions and start two simulations in different, with different initial conditions. These two graphs here are one simulation, population densities of the three groups and the substrates, 4 substrates. This is another simulation. This simulation was started with high initial abundance of the sulfur bacteria. The sulfur bacteria get the advantage here, the home advantage, the starting advantage, and they never give it up because they're at high abundance, they inhibit the cyanobacteria, and so they stay in high abundance, and the cyanobacteria just lose, unfortunately, and the system stays anoxic. Oxygen concentration is very low. Whereas if we change nothing else, but just start the system in an oxic state with high abundance of cyanobacteria then they stay in high abundance and the sulfur bacteria decrease, and we end up with quite high abundance of oxygen. But it's just really this is just demonstrating that that system can show alternate stable states. That's one way of showing it. This is another way of showing it. On the x-axis here, we've kind of got an environmental driver. This is how quickly oxygen can diffuse into or out of the system. Rather inaccurately, I would say we could think about how much oxygen is in the system here. And here we're pushing a lot of oxygen into the system. And actually, there's only one state here, and that is with high cyanobacterial abundance and low sulfur bacteria abundance. When there's very little oxygen in the system, you can only get one state, low cyanobacteria and high sulfur. So this is the anoxic state. This is the oxic state, driven by an environmental driver that we can just force ourselves. It's external. And in the middle region here, we see what we already saw on the previous slide, which is that we can get either state depending on the history of the community, the ecosystem. And so if we start oxic, go along here, follow this solid line until we get to rather low oxygen diffusivity, and then it switches to the shifts to the anoxic state, goes back along here. If we then increase oxygen diffusivity, it goes along. It doesn't flip back here, it only flips back here to the oxic state. Any questions? All right, you've seen that already. So what we thought, let's add some diversity to this. How are we going to do that though? I had a look in the literature and found out that at least the sulfur bacteria can evolve tolerance to oxygen. So maybe tolerance to oxygen of the sulfur bacteria and tolerance to sulfide of the cyanobacteria is a type of diversity that would be important here. So that's what we added. We added, first of all, we made there be 9 strains. Doesn't really matter if it's 9 or 5 or 12 or whatever. Multiple strains per functional group. Before there was only one, now there are multiple. We made them vary in their tolerance. Here, the cyanobacteria's tolerance to reduced sulfur, or the tolerance of the sulfur bacteria to oxygen. We created some variation there. And then we also made there be a trade-off with growth rate. So if you have high tolerance, you have lower growth rate. If we didn't make the trade-off, then the more tolerant one would always dominate. So we had to have the trade-off. And here you can see the result of that trade-off. So as along a gradient of reduced sulfur concentration, we see that the strain with the greatest tolerance has higher growth rate at high sulfur concentration, but low growth rate at low sulfur concentration. It's just a result of that trade-off here. It's just showing it a different way. So we put that variation in, and we also made it so that we could vary the amount of variation. And we had one simulation where there was no variation among the 9 strains. So all clapped down, so 9 strains were identical. In that situation, we should get exactly the same results as when there's one strain with that same tolerance. Oh yeah, here's the equations. There you go. Not only equations, but code as well. What more could you want? These are the equations for the 3 functional groups: cyanobacteria, phototrophic sulfur bacteria, and sulfate reducing. And they're the 4 substrates. And yeah, we did code it in R. It's highly inefficient when we run it, but it's fast to write for us anyway. So if you want to have a look at any of this, there's a really nice supplement with all of the information about how to run this and so on. It's all in an R package, so completely reproducible, which we're actually very proud of. we got a tick or a badge on the paper as well that says reproducible as well. So I should wear it actually more, shouldn't I? I mean, honestly, I don't know if it was worth it. This is something that would be good to talk about because it was so much work to do that part of it to make it reproducible. But I'm not sure where it's showing up apart from these badges on the front of the paper. Anyway, it's something to talk about. Okay, here are the results for when there's no variation among the strains. You can only see one strain dynamics here because they're all on top of each other. High cyanobacteria abundance at the start because oxygen diffusivity is high. This red line is the oxygen diffusivity. So this is what we change. We're forcing the system by decreasing oxygen diffusivity and then increasing it. And so the system should flip from oxic to anoxic to oxic, and that's exactly what happens. Oxic, anoxic, and back to oxic, okay? Nothing new here at all. The question is, that we're trying to answer here, if you remember, is what happens to the position of these shifts when we put some diversity in? Is it going to delay the shift, increase resilience, or is it going to accelerate the shift, decrease resilience? And also that shift back as well. Well, here's what happens. Now you can see the 9 strains because there's variation in the strains in the tolerance. Highest tolerance is dark always. And so you can see that while there's high oxygen diffusivity, the least tolerant strain dominates because it's got higher growth rates. And the more tolerant decrease in abundance, but then at some point, there's replacement of the strains almost simultaneously here towards the most tolerant. And then even the most tolerant can't hack it and dies off. And here is the sulfur bacteria with the most tolerant increasing in abundance first, as one would expect. So pretty much what we'd expect. But now what we have to do is flip back between the previous slide and this one, so we can see what's happened to the shift. Can you see it? It's easier actually if you look at the bottom graph. So this is no diversity with diversity. No with, no with. They shift both that way, right? So they're both delayed. So adding diversity delays, adding diversity increases resilience. Okay. And that's when we add diversity simultaneously to all of those trophic groups. Another way to view this is like this. Here we add diversity from left to right. And here is the effect of that diversity on resilience. This is what we saw before. The shift from anoxic to oxic is happening later, so it's higher resilience. There's a positive effect on resilience, and also the oxic to anoxic tipping point is delayed as well. So there's a positive effect of diversity on resilience there. Do we always see that though? The answer is no. It depends what, don't say it depends, right? Supposed to be able to predict this as well. I think we could predict this from the network diagram if we'd have thought about it a lot before. But of course, modeling, we just did the models and found the answer. We can see that with variation only introduced into the cyanobacteria, the shift to the resilience of the oxic state is higher. So the shift to anoxic is delayed, but there's no effect on the shift from anoxic to oxic. And actually that's pretty sensible because the cyanobacteria are at very, very low abundance when it's anoxic. So what can they do? With variation in the sulfate reducing bacteria, we see the story is not that simple because they have an effect on both shifts, even though they're in low abundance in the oxic to anoxic shift. They do cause a decrease in the resilience of that. And then we get this very strange result, which is that increasing the amount of diversity in the phototrophic phototrophic sulfur bacteria actually decreases the resilience of their own state, right? So they're dominating that state, but adding diversity to them actually causes that state to become less resilient. So that's a negative diversity stability relationship. I better finish. So, and if we think that's because of the complexity in this system fundamentally. So the phototrophic sulfur bacteria consume sulfide. The sulfide is inhibiting the cyanobacteria. So we think, we're not totally sure, but we think that it's that's causing this. So when the phototrophic sulfur bacteria become diverse, more tolerant, and so at higher abundance, more sulfide is consumed, and therefore the cyanobacteria are actually released from some of that suppression from sulfide. I don't totally buy that. I don't think it's the complete story, because when these are at higher abundance, there should be more oxidized sulfur, which should increase the abundance of these. So it must be that there's more of an effect of the phototrophic sulfur bacteria on sulfide and going around this way. Anyway, that's what we said we thought might be happening in the discussion of the paper, but we don't know for sure. So greater diversity can delay a shift. It might not aid a recovery, and diversity in a functional group that co-dominates a state might actually facilitate its collapse. We found all of those three things in there. And it's because this is actually quite a complex system. And so since then, we've simplified this down, and we don't then get such a diversity of effects of trait diversity on the resilience. All right, well, that's it. Okay. Thanks. And there's just the take homes, all repeated. And of course, thank you very much to all of the people that were involved in this research. You've seen many of them there. There were others as well. And all of you for listening. You clapped a little bit too early, but please don't clap again. Just some questions, maybe. Dave?
Yeah, really interesting last results. I hadn't seen that paper. Having worked on some of these purple sulfur and cyanobacteria, I can't imagine doing the experiment then, given how slowly they can grow.
So we tried.
Yeah, how'd that go?
I mean, the first thing we did was we made some Vinogradsky columns.
Oh, right. Okay, sure.
And if you know, these are glass columns, and you put in some sediment, and then actually put in some nutrients that contain sulfur, like egg yolk is a good one. And then over time, you get these layers of beautiful different colors of green, and then purple and brown. Quite a lot of museums have them, actually. Anyway, we made some of those, and then we extracted some of the organisms from them. And did quite a few experiments. I mean, the only thing that we were trying to do at that point was actually show that we could get alternate stable states and recover those kind of dynamics. And I think actually we didn't demonstrate it unambiguously in the lab. We got close enough to submit a proposal, but well, actually not close enough to get it funded. But none of those experiments involved manipulating trait diversity. That was going to be the stuff that we did in the work that we were trying to get funding for.
So I guess I was just wondering, what I found really interesting was, I guess, the delay in the catastrophic shift with intraspecific trait diversity, right? And that delay in itself, I mean, from your sense and from how the equations work, like, could that then also be interpreted as a time lag, which could then either actually worsen the stability given slight changes to the model, right? Because I'm thinking, you've got these like continuous time differential equations. You have any sort of competitive interactions between these things that could then end up like destabilizing or making the catastrophic shift a lot worse, just based on like the sensitivity of the parameters.
Right. So
You have a shiny app that you can.
no, nothing so cool. Yeah, this one. So
I'm thinking when you switch between those two scenarios,
maybe this,
yes,
yeah.
right.
So this is a little bit, what do I want to say? A little bit of a distraction, or because this is how I'm showing the results to you, but the actual calculations of the effects on resilience were done done in a different way. So we ran the model for a long time to get the final state. So in the resilience results, there's no time, there's no possibility for kind of time lags to be involved. Whereas here, there is. So actually so what I'm trying to say is the level of oxygen diffusivity here that we're seeing the flip at might not be the same as in those stable state results that I showed, because it does take a little while. We have not done what you're talking about, which is look at the effects of faster changes in oxygen diffusivity. If it happens too fast, we could end up with the system still in the oxic state, even though it should be in the anoxic state, right?
Yeah.
Which is gonna.
There was some similar work with, I think Aaron Ellison and company wrote papers with pitcher plants where they kind of artificially changed the environment from oxic to anoxic. And it worked really well because all you have to do is feed carbon into the plant, and through the bacterial communities and whatever's in there will turn the state anoxic. And they have some cool models that are pretty surprising.
Conscious of time, maybe one quick question if someone has one.
So if these experiments, Petri dishes or Winogradsky columns are small buckets, what kind of a bucket is our code?
Infinite.
Infinite bucket, infinitely big.
This one is, right? Because this is no specified volume, it's just density, concentration. So I guess that's an infinite bucket, deterministic.
Well mixed.
And well mixed, yeah.
You had temporal variability in oxygen diffusivity. Could you see flickering between alternative stable states? Right now we're treating this as just a continuous grind way. I'm trying to think of like, is there a system that would just bounce back and forth between those? What would that look like?
So would we see flickering? We've forced this system with some continuous fluctuations, and we didn't find any evidence that these early warning signals were predictive.
That's fair.
Yeah. But we couldn't get it, we never got it to flicker between the two states with that specific fluctuation we were making.
And so in that hysteresis zone, if you had enough like of a push, could you get it to bounce back and forth?
I think we'd have to push it quite far, and we'd have to leave it for a while as well. Okay. I'll just continue talking. Please make me stop, right, when you want to leave. I was saying that we did, before we did all this work, I made a reproduction. I got into this kind of habit of reproducing people's work in R. Right? And so I reproduced that model of Bush, and I couldn't get the same results as him. And I managed to track him down, and I said, what's going on? Give him my R code, and I said, what's going on here? And he said, oh no, you've written everything correctly. You're just not running the model for long enough to get to the stable state. And I was like, okay, so how long do I have to? And this was not running. This was a number of this was biological time. So I said, well, how long do you have to run it for then? Because it was quite a long time already. And he said, well, you need to run it so biological time of like several years. What? Does that make any sense? Because this is a study of lakes and mixing in lakes, and that's on an annual basis. So actually, by doing that reproduction, I found something that was not in the original paper, but I think actually pretty critical, and that is that the temporal scale that's required to get these types of results is a very questionable, has very questionable relevance to the actual real system that they're talking about.
500,000 hours is more than 10 years, isn't it?
Yeah. So reproductions are important. So what I learned from doing quite a lot of reproductions is it's quite useful to do them. One does learn things, but actually it's very difficult to communicate those things in a positive way. And so I didn't do anything with it really. I'm just saying it now. I think the study is really, really nice one that they did. Yeah. But fortunately, we got it published without anyone complaining about that as well. So there you go.
Okay, well, thank you very much, Owen. And this series of talks will continue on Monday afternoon and then Wednesday afternoon next week. Thanks for coming.
I was just struck by, I remember now. You know, this person, no, this is because Will then about Santa Barbara. The sulfur cycling sorption only found in Massachusetts by Koch on the and she treats them as if they're, so they basically sulfur oxidizing or sulfate reducing bacteria. And then of course there's cyanobacteria. Okay, these little, they call them areas. She's using them as microbiology.
I thought there might be some stuff, but there wasn't.
The lab.
Yeah, so you can bring it into the lab.
We just assume like
Like,
everything,
no,
category dies.
Yeah. The same rate or the same.
Yeah, the evolutionary responses are actually really important.
Yeah. I know, the first Sunday afternoon, I wouldn't mind.
Yours is more.
And she.
It's also.
It's one of those, really interesting little that, you said it, the question.
It's like,
Zooming on back of the study.
Yeah, it might be hard to find the right like noise model and strength that produces it, but I don't think it's, but I don't think it can be the case that don't feel like. They completely didn't
All of that, so the co-star Chloe Ash is looking at community environment now, which is.
Okay.
And I know she talked to me before, I texted her and all like.
Yeah.
That's because she wants to look at like.
So I actually wrote a paper, but this is very just Volterra competition is the.
I'll look at what it's been doing.
And it was with Priscilla's tacos, and we were interested in whether we were calling it the sweet spot. So not enough noise and you never switch. Like, so let's say we're not even moving along that bifurcation diagram. We're just in the alternative stable.
Typhoon related things like I know.
Too little noise and you just stay at one. You know,
Yeah.
the second stable states are relevant. And too much noise and you're just seeing the signal of the noise and the existence of any stable states is irrelevant. And so where you get this flickering is the sweet spot, and we looked at various models with either two stable states or a long transient and stable state or something, and found the sweet spot by some artificial definition that was created.
Let's do that.
And we tried to find signal in that sweet spot, like is the noise,
Yeah, that'd be great.
It's new for her. So
does it tell you whether you really have two stable states or a long transient? Like, no, it tells you absolutely that. We could not find anything that was, so we wrote a paper about how like,
Cool.
look guys, long transients are just as relevant as steady states. Well, they are steady states, they're just not even stable equilibria. Yeah, anyway. But I think we must get flickering in anything. Just may not be ecologically meaningful. well, yeah, to kick it.
Oh, yes.
Yeah, sweet spot of kicking something.
Yeah, it's been a hell of a lot.
How do you get?
Feels like beer
Is it
time.
like-
Yeah. Scholarship theoretical ecology. There's a special issue that Alan Paddington and I put together on.
So well.
I was so surprised. I mean, it's expensive.
It's in there.